1 in 4 children born today will live to celebrate their 100th birthday. In fact, some scientists think that a baby born today will live on average 5 hours longer than one born yesterday. So the question is, could you live forever? And would you want to? Animals like this lobster live for an extraordinarily long period of time. So what do animals like this have to reveal to us about the secrets of eternal youth? Institution 2013 Christmas Lectures. My name is Alison Woollard and I'm 437 years old. Today we're exploring Life Fantastic. But before we start, there's just something I have to do. Some of you seem shocked that a 437-year-old could backflip through the lecture theatre. Well, do you know what? That wasn't actually me. And I'm not actually 437 either. But if we wanted to live forever, that's the kind of thing we would all want to be able to do. Because we would not just want to be older for longer with all the diseases that we could get in old age. We would want to be fit and healthy for longer too. But now I need to introduce you to Francesca, my stunt double for this evening. I like the hair. Thank you, Francesca. And today, that's what we're going to explore. How what we're learning about how our bodies age and how they become diseased could help us live healthier lives for longer and explore just how far down the road of living forever we could go. If we want to extend our lives, to be healthier for longer, perhaps, who knows, even live forever, we first need to know about life itself and natural lifespan. We all have a lifespan. We're born, have a childhood, had teenage years like many of you are doing at the moment. We mature, we grow old, we die. It's inevitable. But not all animals have the same lifespan. In fact, animal lifespans are hugely variable, from just a few minutes to hundreds of years. Let's look at this in more detail, shall we? Can I have 2 volunteers, please? I'll have you there in that row, and I'll have you in the blue jumper. Thank you very much. If you'd like to come down. If you'd like to come round to the back here. Hi. Thank you. If you'd like to come down as well. What's your name?
Thea.
Thea, and you're a geek, are you? We like geeks here. And what's your name?
I'm Kelly.
Kelly, hi. Now, we've got some animals in front of us. You're not frightened of animals, are you? No. No. Should we find out what they are? Yeah. Why don't you lift the cover on this one, and we'll see what we've got. Ah, what do you think they might be?
They look like mussels.
Mussels, yeah. They're actually clams. Clams. What about this one? Have a look in there. Oh, what's that?
Turtle. A tortoise.
It's a tortoise. Isn't it sweet?
Yes.
Do you want to have a look in this one? See what's in there. Oh, look at these lovely little hamsters. And what about this one? What's in there? It's a lobster. And what we're going to do is we're going to try and guess their ages. Okay? So we've got some cards to help us do this. And we have 3 cards for each animal. Okay? And we're going to start with this animal on the right, with the lobster. So we have 3 cards to choose from, 3 lots of years. So we have 5 years, 50 years, or forever. What do you think?
I think 5 years.
5 years?
I think 50.
50? What does the audience think? 5? Who thinks 5? Who thinks 50?
50.
Who thinks forever? What do you think? What do you think? What should we do?
I think because of the audience, 50.
What do you think?
Yeah, 50.
50, should we go with 50? Right, we'll put that one there. And the next one, these little hamsters. We've got 3 cards for them as well. So their lifespans could be 2 years, 5 years, or 10 years. What do we think?
2 years.
Have you had one as a pet?
No. But
No. What about you?
2 years.
2 years. Let's go 2 years. Okay, the next one, the tortoise. Now, what's the choices here? We've got 50 years, 150 years, or 300 years. So what do we think with a tortoise?
I think 150 maybe.
100? You reckon?
Yeah, 150.
What, higher? Lower? What does the audience think? Higher? Who's for higher? Well, not many. Who's for lower? Oh, I think we're all agreed on 150 years. So we'll put that one there. Don't eat it, will you, Mr. Tortoise? Right, and the last one here, these clams. Oh, gosh. Big range. 5 years, 100 years, or 500 years? What do you think?
I think 500.
500?
I'll go 100.
You go 100. What do the audience think? 500? Who thinks 500? Oh. Who thinks 100? And who thinks 5? Looks like we're going to go with 500, doesn't it? Let's go with 500 over there. Okay, now let's think about how we did. Actually, you know what? You're very good. You're pretty spot on with a lot of these. So the lobster does indeed live for about 50 years. I mean, some reports that suggest that lobsters are actually immortal, but they're probably not. They're probably not. And most lobsters will live for about 50 years in the wild. There's some that will get a lot bigger than that and will live for a lot longer, but the average lifespan is about 50 years. And our little hamsters, they've gone to bed now, haven't they? But that's right, they live for about 2 years. Most rodents of about that size will live for about 2 years. He's coming out to say hello. He's very sweet. Actually, if they were in the wild, they'd probably live a lot less than 2 years. You think why?
Because there's more chance that they'll be eaten by other animals.
They'd probably get eaten. Yeah, they'd probably get eaten. And the tortoise, well, some tortoises do live a lot longer than 150 years, but 150 years is the average age span. In fact, you know, there are tortoises alive today who were already 10 years old when Darwin published his book, The Origin of Species, in 1859. Isn't that amazing? And the clam. Do you know, I don't know, you're absolutely right that some clams can live over 500 years. And have you read about that recently?
No.
And in fact, scientists have found a clam that they think is about 570 years old, and that's supposedly the oldest animal that's ever been found. These particular clams we have here, this type of clam, probably lives more like 5 years. But we can see a huge variation in these lifespans, can't we? Thank you very much for helping me with this demonstration this evening. So the truth is we really don't know what controls animal lifespans. It's down to a huge number of things. And I think we need to explore this by starting with us. I want to show you something now. I want to show you a whole lifespan in a few minutes. This is a video that records the whole lifespan of a particular person, speeded up. It's very seamless speeding up, and we'll see this person age. And I'm going to leave this running, it goes on for a few minutes, and as it's running, you'll start to see changes happening in this person. It doesn't happen overnight. In fact, it's an extremely gradual process, one that we barely notice. And I'm going to leave this running in the background. But if you suddenly look at this and think that person's got old, I want you to tell me. So shout out when you think this person has just suddenly got old, and we'll try and work out what it is that makes someone age. But could you imagine if you could tell what you would look like as you got older? Imagine if we could look into the future. What do you think you'd look like when you're older? Do you think you might look like your mum or your dad or your grandpa? And would you change it if you could? Well, we've got some software that means that we can tell what you'll look like when you're older. So, shall we have a look? Who would like to have a look? Right, now we've actually got some photographs that we elicited from you earlier on. And the first one will come up. Is this person here in the... it's you, Amir. Amir R. Is that you?
Yeah.
Right, and this is you at 13?
13.
You were 13? Is that a recent photograph?
Yeah.
So you're 13 now?
Yeah.
So should we see what you look like when you get a bit older? Are you ready for this?
No, but for the purpose of the lecture, yeah.
We'll see. Right, let's age Amir, please. So 14, 15, 16, 17, 18, 19, we're getting a bit older. It's looking all right so far, isn't it? 30s, 40s, you're doing all right. 50, what's happening? Your hair's going great. 72. Well, I don't think you look bad for 72. I mean, what do you think?
It's really scary.
Is it scary? Do you look like one of your older relatives, perhaps?
Yeah, like my
You like your
granddad.
You're like your granddad. Well, I think he looks pretty good, pretty wise and very nice. So that is a glimpse into your future. Shall we have a look at another one? Natasha. Is over here, Natasha. You look a bit apprehensive. So this is you at 13. So is that a recent picture?
Yeah.
All right. So should we age you?
Sure.
Yeah. You ready?
Yeah.
Right. Let's go for it. You're moving on. 28, 33, 40s, 50s, 60s. Hair's going great. 70s. Skin's going a bit saggy. 72. What do you think? What do you think?
I can wait to get old.
You can wait. You don't want to do it tomorrow?
No.
No. The person's age, yes, they have, haven't they? They do look significantly older now. You can see lines. Do you know when that happened? Could anyone put their finger on exactly when it happened? Very hard to put your finger on the precise moment, isn't it? Because so many different things are happening to that face at once. It's a really clever bit of film. So if you could slow this process down, then would you? So we got to the end of our video with Danielle, and we saw that it was a very, very slow process indeed. So what's the biology behind this ageing business? What biological processes are going on to influence the way that we look and the way that we respond as we get older? So it turns out that we have a lifespan because our cells have a lifespan. And each different type of cell has a different lifespan. And we need a body to look at this, but not one of you. We've got a special body all of our own called James. James, hello. Nice to meet you. And what we're going to do with a very special projection is we're going to dim the lights in the lecture theatre, and we're going to use some special projections to actually look inside James's body. Wow. That's beautiful. And so we can see his different tissues and organs and cells. And we can start to think about the different sorts of cells he might have in his body and how they might age. So one of the most prominent tissues we can see in James is his vasculature, his blood cells. And our red blood cells would last typically for around 4 months inside our bodies. And then we can see his liver. And our liver cells in adult humans will last for over a year. Actually, the liver is one of the most easily regenerated organs in the body. We can make lots of new liver cells very easily. And if James turns around, I think we'll be able to see his nervous system here projected. Wow, look at that. You can see all the neurons in James's body. And our neurons are very long-lived indeed on average. In fact, most of them are there from when you're born until when you die. Although not all of them. Some neurons do have a much shorter lifespan, but most of our neurons are there with us for our whole lives. Okay, James, thank you very much for showing us your body. I'm just having a bit of a scratch, and I think you should all scratch your heads as well. Can you all scratch your heads for me? Well, when you're doing that, you're actually losing thousands of skin cells. In fact, you lose about 500 million skin cells every day. And your whole skin, all your skin cells are recycled every 2 to 4 weeks or so. Okay. Does anyone have a dusty house? Does anyone live with dust? I do. You're not wanting to say because you feel you might embarrass your parents, but come on, does anyone have a dusty house? Yeah. Well, that means you're shedding lots of skin because dust is composed primarily of dead skin cells. So our skin and most of our cells need to be constantly replaced. In fact, by the time I get to the end of this sentence, several million of our cells would have died for one reason or another. All this replacement's a funny business when you think about it, isn't it? It's a bit like this broom. Thank you, Sophie. This broom. This place could do with a bit of a sweep. Do you know, I've had this broom for about 20 years. It's had about 5 different heads and had about 6 different handles, but it's still my broom. That's a funny thought. If you replace the head because it's worn out, the head of a broom, and then the next year you replace the handle of the broom, is it still the same broom? Well, if most of the cells in your body, let's leave our neurons aside, if most of the cells in your body are less than 10 years old, you always think of yourself as the same person, don't you? But are we? Our cells are being constantly replaced. Interesting thought, that one, isn't it? Thank you, Sophie. Now, as we get older, the cells responsible for regeneration stop playing the game, and regeneration slows down. And one of the ways regeneration slows down is that the cells that are meant to be regenerating just stop doing the things that they should normally be doing. They shut down. And that's what we call cell senescence. But if cells stop regenerating as they get older, then they must have some way of knowing how old they are. So how do cells know that they're getting old and they really ought to stop doing what they're doing before they start to do something really wrong? They must have some sort of counting mechanism, some sort of clock. And it turns out that that's exactly what they do have. I want to show you something now. I'm going to show you a picture of a cell with some chromosomes in it. And those chromosomes are splitting apart as the cell divides, as we can see here. A chromosome is a linear molecule of DNA, and all the DNA in our cell, the genome, is divvied out into these linear chromosomes. We have 23 pairs of chromosomes in each of our cells, and every time a cell divides, the whole chromosome has to be replicated. And when a chromosome is replicated, some of the genetic material gets lost from the ends of the chromosomes. It's just part of the machinery by which DNA is replicated. It's not very good at replicating the ends of the chromosomes. And so this can be bad, as genetic information can be lost from the ends of chromosomes. It's a bit like thinking about a chromosome as a shoelace. Kim, come here. I've been looking at your shoelaces for the last few minutes, and I can see that they're in a really bad way, aren't they?
Sorry, yeah.
Fancy coming on TV with frayed shoelaces?
Sorry, Mum.
Yes. Don't they pay you enough round here?
No comment.
No comment. So you can see that this shoelace is horribly frayed and it's all going to fall apart, isn't it? So what would have kept that shoelace nice and neat? What would stop it fraying?
Those little plastic bits that they put on the ends.
Those little plastic bits that they put on the end. And I've just spotted some nice little plastic bits on the end of some very fine shoelaces on Carolina. So let's have a look at your shoelaces. No fraying. Do you know what the end of a shoelace is called?
No idea, actually.
No idea. It's called an aglet. So you have aglets and you don't have aglets. So your shoelaces are nice and neat and yours are frayed. And it's just like that. Thank you, you two. It's just like that with chromosomes. Chromosomes have special protective caps on the end of them too. But they're not called aglets. They're called telomeres. We've got a model of one here. In fact, we've only got half a chromosome. We were going to put a telomere on the other end of our chromosome too. But then we did some little calculations and we worked out that the scale we have of our representation of a chromosome is a piece of rope. The width of this chromosome would mean that it would need to stretch all the way from here to Madrid if it was drawn to scale. And we couldn't afford enough rope to go from here to Madrid. So we only have a chromosome with one end. So this protective cap on the end of a chromosome is called a telomere. And a telomere is made up of repeating units of DNA. And so as the DNA replicates, then bits can fall off the end of our chromosome. So units will come off every time a cell divides. And it's not too much of a problem because these telomeres are just these repeating units of DNA. But we will get to the stage where the cells started to recognise that maybe this isn't such a good idea if it carries on. And we see we got to sort of red alert stage here. So if we remove the next one, I didn't know that was going to happen. Okay, that's enough. We've got the hang of it. We can see that things have become disastrously short for this chromosome. So the cell recognises it's too short and it shuts down and it stops dividing. This process is called cellular senescence, and it's thought to be one of the important mechanisms, there are others, but one of the important mechanisms controlling the lifespan of the cell. And it's thought that senescence probably contributes to the lifespan of an organism as well. So although the telomeres protect the chromosome from information loss for many replications, there comes a time when enough is enough and the cell shuts down. And that's one of the things that contributes to our aging. It's all a bit depressing, isn't it, this? All sounds terrible. All this talk of cells shutting down at the end of their life and possibly the end of an organism's life. But it turns out that there are other mechanisms of cellular shutdown. And one of these is actually crucially important at the beginning of life. Let me show you another video. This is a different kind of cell shutdown. This is cells dying, but we can see that they're going to do it in a very spectacular way. All right, we can see this is a cell going out in a blaze of glory. It's an active self-destruct mechanism, and it's really important. It's called apoptosis, or programmed cell death. Have you heard of apoptosis? A few nods, not many. And it may look bad, but cell death like this is actually part of building a healthy body. Weird as it sounds, cells must die as part of the embryonic development process. Take our hands and our feet, for example. We've 5 digits on each of our hands, haven't we? Perfectly formed to grasp, gripple, and manipulate. But how did they get this way? Well, it turns out that in our development, when we're developing in the uterus of our mothers, the human fetus has webbed fingers and toes. And it's this important process of programmed cell death, or apoptosis, that kills off the cells in the webbing. And this is part of the normal developmental process. So it's crucial for some cells to die during development. So what would happen if this process of programmed cell death didn't happen properly? Well, let's see, shall we? I need 2 volunteers to help me with this. Yes, we'll have you, and we'll have you on the end there. Now, if you can come and stand here. What's your name?
Ben.
Ben. And if you can stand here, what's your name?
I'm also Ben.
Oh, no. Well, that's easy for me, isn't it? Ben and Ben. Now, are you good at cutting out?
All right.
You're all right. Right. Now, what we're going to do is we're going to cut out these shapes with these scissors. And you're going to start here, and you're going to cut all the way around here until you get to there.
Okay.
Right? But you're not going to cut that bit, and then everything will be nice and sticky togethery. Right? Now, you are going to do the same thing. But first, you're going to put on our little webbed gloves.
Okay.
With the help of Lin here, because they're a bit of a squash. Okay? Right. We're not going to start quite yet. So we're just going to wrangle the gloves onto the Ben.
Done. Yeah.
Gosh, that was quick. Right, now, next one. Get your fingers straight. That's it. Make sure one's pointing into each fingery thing. Give a good tug. How's that?
Good.
Weird, huh? How's it feel? Feel
weird.
like a duck?
Yeah.
Right. Here's your scissors. Here's your thing. You're going to cut out. You're going to cut all around the fingers here. Okay. And then we're going to cut out some hand shapes. Right, are we ready to go? I'm going to say, on your marks, get set, go. Right? On your marks, get set, go. It's a race. So go quick. Okay. Oh, my word. Do you need a bit of help? I'll tell you what. Should I hold this?
Yeah, that would
And you
be
hold
great.
the scissors. Okay. And we'll work together. Ooh. I think Ben number 1 over there is... Oh, it's a work of art, this. Oh, dear. Oh, hang on. We can let you... I think we can let you have a... Anyway, I think you're on your own now. You know what you're doing with these gloves. So it's not too bad, is it? Come on, this is meant to be a race. Come on, bit of urgency. Oh dear, showering me with bits of paper. You're starting to get the hang of those. Starting to get the hang of those gloves. Okay, right. I think we'll stop there. That's absolutely fantastic. Almost. Brilliant. Now you have got a nice... Oh, look at that. Isn't that lovely?
Yeah.
You did that all by you can keep that. And what? Oh, I'm wrong with that. It was hard, wasn't it?
Yeah.
It was hard enough for Ben, and he had all his fingers. The goodness knows what it was like for you. So was it a bit of a handicap?
I think so, yeah.
I think so too. So if you didn't have this programmed cell death going on and removing these bits between your digits, life would be a bit more complicated, wouldn't it?
Yeah.
I think we've made the point brilliantly. Thank you very much for helping. So we can see how awfully difficult life can be when we don't have programmed cell death happening at the right place at the right time. But it turns out that apoptosis is not just important in development, it's also important for maintaining a healthy body. In fact, it turns out that billions of cells in our bodies undergo apoptosis every day, and we don't even notice, do we? Can anyone feel apoptosis going on inside them at the moment? I can. I don't know about any of you. So apoptosis is a good thing. But when the regulation of apoptosis goes awry as we age, this can mean two things. So if the control of apoptosis goes wrong in such a way that there's too much of it, then what will happen? We'll end up with fewer cells than we should have, shouldn't we? We'll end up losing too many cells. And scientists think this contributes to the loss of neurons in neurodegenerative diseases like Parkinson's and Alzheimer's diseases that are associated with age. Too little apoptosis, on the other hand, and well, should we do an experiment to see what happens if we have too little apoptosis? So what we have here coming on, believe it or not, is a body. A low-budget body. This box represents a body, and as we know, bodies are made up of cells, and the cells are these little pink balls in the middle, okay? But our cells need to regenerate themselves and proliferate and so on, so we need some spare cells. Thank you, Sophie. So we have some spare cells here. And these are our normal cells that do what they're meant to do and behave well. But we also have some abnormal cells coming on, and they're blue. And these are cells that might accumulate in our body and have a bad outcome. Now, it looks to me like I'm going to need 3 volunteers for this experiment. So let's have you there in the blue T-shirt, and we'll have you in the middle there, and yeah, you two. Yeah, that's great. Thank you very much. Right. Now let's have a look at you. I need to assign tasks. Right, we'll put you over here just for a minute. All right, we'll come back to you later, and we'll have one of you here. Right, and we'll have one of you here. If you just sort of get in that little gap there. And what's your name?
Louise.
Louise.
And Kieran.
Kieran. Louise and Kieran. Now you two are in charge of cell proliferation.
Okay.
Can you manage that? Right. So what you need to do is add cells into the body. Okay? So that's supposed to represent cells making more of themselves and dividing and so on as we grow and develop and have a life. Okay? So what you're going to do is basically put the cells in one at a time as fast as you can into our body, but not great handfuls at a time, because that would be cheating, okay? And now we have our very important volunteer coming to stand in the middle here. What's your name?
Gwynval. Sorry?
Gwynval, actually. Gwynval, but it's not, I'm afraid. For the purposes of our game, your name is P53. Easier for me to remember. Okay, so we've actually got a sash for you that says P53. You just put our sash on somehow. Okay, we got P53 sash. Oh, it's upside down. That's no good. Other way up, Kim. Other way up. Dearie me, you can't get the staff these days. Right, P53. Excellent. P53, ladies and gentlemen, is a very important control protein, because P53 seeks out and destroys abnormal cells from the body. And that's the job of our P53 protein here. So you are in overall control. Your job is, as soon as you see a blue cell coming into the body, you take it out and you put it in the box behind you. Okay, now it's much better if you all kneel down, because it's easier for you and it's easier for our cameras as well to get it all. So we're going to run the experiment for about 10 seconds, and I'm hoping that the audience is going to help me with a countdown. Are you prepared to do that? Help. Good, right. So, are you all clear on what you're going to be doing? Right. On your marks, get set, go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Stop! Stop! Right. Very good. Thanks. So now, overall, I would say that our P53 hasn't done a bad job. He's keeping control of the situation. There's a few of those dodgy cells in our body, but not many. Most of them have been taken out and put in our box. So I would say P53 is in control and things haven't gone too badly wrong yet. Now we're going to run the experiment again, but this time we're going to give P53 a mutation, right? The P53 gene is going to have a mutation so that the protein won't work properly. Now don't worry, we're not going to chop your hands off, but we are going to make you wear some boxing gloves, so that it may make it slightly more difficult for you to remove these bad cells from our bodies. Okay, boxing gloves on. Excellent. Okay, so we're ready to play the experiment again. Right, on your marks, get set, go. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Stop! Not bad. Not bad. Are you a boxer? You seem quite good with those gloves. Maybe you should take up boxing. But we can see that things have started to get a bit messy, haven't they? You've removed a few of the wrong sort of cells I see there. But also we can see that the blue cells have started to accumulate in our bodies, haven't they? Just bring the camera in. You can see that there's much more of a build-up of these blue cells. And so that might actually be a tumour forming in a body, mightn't it? Okay. Thank you so much for helping with that demonstration. Fantastic. Thank you. So this experiment then has shown us that if your P53 protein becomes impaired, you're more likely to get cancer. And P53 can stop working properly because it acquires mutations. And the older you get, your more chance your P53 gene has to acquire mutations, because mutations can happen throughout the lifetime of a cell. So in a nutshell, living longer has risks. So can you have your cake and eat it? Can you live longer and not start to wear out, not be more likely to get sick? Well, now I want to introduce you to an animal that seems to have cracked it. An animal so special it lives at least 10 times longer than you would expect. And not a single one of these animals has ever been found with cancer. And they're coming in now. And I could do with a volunteer to help me look at them in more detail. Oh, let's have you there on the end. Thank you. What's your name?
Ella.
Ella, right. And come round the back here and let's have a look at these little rodents. Aren't they cute? Look at their little teeth in the front. Well, they're quite big teeth, really, aren't they? Not sure I'd want to stick my finger into one of those tubes. So what do you notice about them?
They've got no fur.
They've got no fur. That's right. And what do they look a little bit like?
Moles.
They do. Do you know what they're called?
Are they mole rats?
They are naked mole rats. Well done. And naked mole rats are extraordinary animals because they live for such a long time. Thank you very much for helping me identify those animals. So, these naked mole rats might not look that pretty, although actually I think they do look absolutely gorgeous. Who thinks they look lovely? Yeah, I think we're all fans of the naked mole rats in the Royal Institution this evening, aren't we? They're really, really lovely. And they've cracked a problem that we haven't. And so studying them may well give us amazing insights into ageing and also how to beat cancer, because no one has ever found a naked mole rat with a tumour. So you would expect a naked mole rat to live about as long as a similar rodent of the same size, say a hamster, the one that we saw earlier on, about 2 years, but they don't. So naked mole rats produce a very special protein. That's part of their secret. And the protein that they produce is called high molecular weight hyaluronic acid. And we can see it here. It's quite gloopy. And this protein is probably part of the machinery that enables the naked mole rat to be perfectly adapted to life underground. So it seems to contribute to the fact that they have this very elastic skin that they need when they're moving around in these very tight tunnels. But this protein seems to have special properties, and it seems to be associated with making these rodent cells almost completely resistant to cancer. In fact, when scientists take away the ability of the naked mole rat cells to produce this high molecular weight hyaluronic acid, the cells can develop cancerous properties. So scientists don't know exactly how this substance, this high molecular weight hyaluronic acid, works yet. But perhaps we should be really looking at this to unlock the naked mole rat secrets if we want to remain cancer-free and extend our lives. And I'm sorry to say that we're going to have to say goodbye to our lovely naked mole rats now. Such a shame. Thank you so much for bringing those on. So now we know that cells are crucial in determining our lifespan and how healthy we are. But what else? Is it more complicated than this? Well, actually, there's another animal that can help us out here. And this one is very dear to my heart. It's my hero animal, the nematode worm Cenorhabditis elegans, or C. elegans for short. And this is a tiny nematode worm that lives in the soil. It's about 1 millimetre long. And when we grow them in the lab, we grow them on these little Petri dishes. And if you hold these Petri dishes up to the light, you can see tiny white threads on the dishes, and they're the worms, tiny white threads of about 1 millimetre long. And these worms are incredibly useful to biologists because they're very simple. I'm going to pass some around so you can have a look. They're very simple, and they are composed of only about 1,000 cells. And here they are. Here comes Mum, cruising down the centre of the plate. And what we can see is that they're transparent, and that's really useful because it means that you can look at all the cells inside the worm, and there's only about 1,000 in these animals. So that means that we can understand their development very easily by watching these cells and working out where they go. And you can see all the other baby worms around near Mum, and also the oval things are embryos. Okay, but not just important for telling us about the development of animals, C. elegans is also extraordinarily useful for helping us to understand about ageing and lifespan. Because these worms have quite a short lifespan, about 2 to 3 weeks. But we can fiddle around with their genes and we can speed up or slow down the ageing process. And that will help us to understand how ageing works. So Carolina's going to show us some worms of different ages. We've got some plates of worms here. And this worm is about a week old, okay, so quite a young worm. You can see she looks youthful for a worm. You're probably not so used to looking at worms as I am, but this is a nice, youthful looking worm moving around very happily, looking very elegant. And the next worm we're going to look at is much older than that, so about 3 weeks old, and unfortunately coming to the end of her lifespan. And what we'll see immediately is that this worm looks old. Oh dear. She's a bit wrinkly. She's not moving very much. She might move a little bit. Very, very tiny movement, so we know that she's still alive, but we can see that she's really not moving around very much. She looks quite old. Now we've got another worm to show you. And actually, I should say that this is a real experiment because Carolina is a PhD student in my lab. And we've been fiddling around with worm genes, and she's been fiddling around with a particular gene, and we've never done this experiment before. So we're looking to see if this worm lives for longer than a normal worm would do. And this worm is actually quite a lot older than the old worm we were just looking at. But we can see that she doesn't look so old. She's moving around as if she was a young worm. So she's much happier, she's much healthier, and this is because of a mutation in a particular gene. So ultimately what this shows then is that although lifespan is complicated, it does have a strong genetic component. It's written into your DNA. Carolina, thank you so much for showing us these worms this evening. So we know from studying these model organisms like C. elegans that there are some genes that are really important in controlling the aging process. Well, I can see you thinking that's all very well and good for the worm, but what can that possibly mean for us? Well, it turns out that actually we share a lot of our genes with this humble worm. And even some of the genes that are involved in regulating lifespan have a human counterpart. And scientists have even looked at some long-lived people, and they find that some of these people have mutations in similar genes, so have different forms of some of the genes that we also know control lifespan in the worm C. elegans. So that means that studying the process in C. elegans, studying aging in this humble worm, might help us to understand more about the biology of aging in us. That's an awesome thought, isn't it? So we've learned that aging is controlled by our cells and by our genes, and that aging is associated with particular diseases like Alzheimer's, problems with eyesight, higher rates of heart disease and cancer. So what scientists really want to do now is to work out what we can do about all of this. Well, one of the things we can do is to work out how to replace worn-out cells. As an animal develops, it's all about its cells doing the right thing in the right place at the right time. Cells need to differentiate or take on the correct job. So cells follow a program as we develop. And these programs that cells follow are programmed by the DNA. So the fate of every cell is written in the genes. So what if we could intervene with these programs? What if we could wipe it and reset the program? Could we get cells to do whatever we wanted them to do? Become heart cells, for example, to replace worn-out ones, or new eye cells for those with failing sight. Is this possible? Well, to tell us more, please welcome Natalie Mount from Cell Therapy Catapult. Natalie, hi. Good to see you. Thanks for coming. So Natalie, can you tell us a little bit about your work?
Yes, what I do is to translate all this exciting science and understanding we have about cells into how those cells can become new medicines for patients. This is what we call our bench to bedside process, the journey from the laboratory bench to the bedside for the patients.
And you make medicines out of cells?
Yes, that's right. These are a really new type of medicine. They're not like the traditional medicines we think of, where we have chemicals in a pill that our doctor gives us. These are new living medicines, and because of the things they can do, cells have the potential to be able to treat diseases that other medicines can't.
So how do you do this?
Well, we now have processes where we can take cells from our own body, we take them into the laboratory, we grow them, we modify them a bit, then we put them back into our own bodies, and then they can set to work to heal diseased areas.
So what kind of cells are you using for these sorts of therapies?
Well, a really good example is actually in the eye. The eye is a very important organ for us. Obviously, sight is an extremely important sense, but unfortunately, a lot of diseases can lead to blindness. So what we're now doing with cell therapies is to take a little bit of those stem cells from a healthy eye, we can grow them in the lab, and then we can put them back into the diseased eye.
So wait, you take the stem cells from the cornea of a healthy eye, you grow them in the lab, you make them happy, you get enough,
That's right.
and then you put them in the unhealthy eye in some sort of surgical procedure.
That's right, yes. And what happens then is that those cells set to work and can make the cornea go transparent again, so people can see again, and it takes away the pain. This has been a very successful therapy. It's one of the pioneering stem cell therapies.
Natalie, thank you so much for sharing these exciting discoveries with us this evening.
Thank you very much.
Thank you very much.
Thank you.
Natalie Mount. So Natalie was talking to us about therapies which are either available now or going through the trial process. But what of the future? Well, to talk to us about some truly impressive advances, ladies and gentlemen, please welcome Pete Coffey.
Hello.
Hello.
For this guy. There you go. Hi.
Hi. And I understand you're going to talk to us about eyes as well.
I am indeed. As a profession, I'm interested in eyes, but I'm interested in the back of the eye.
The back of the eye, so that's the retina, right?
Yeah, so the seeing part of the eye, you've heard about the window on the front of the eye, but actually the mechanics of vision and seeing, and actually seeing the lecture this evening, is based at the back of the eye.
So what's all this paraphernalia you're bringing with you there?
Well, hopefully it's paraphernalia which is going to keep on. And it's actually, we're going to show you the back of the eye live in the lecture theatre.
Wow.
And we have two guys who are going to help us with that. We've got Nick, Dr. Nick Mathia.
Hi, Nick. Hello, Nick.
And we also have a volunteer, hopefully. Misha, somewhere at the back there is... Oh, yes, we have our volunteer.
Misha. Hello. What are you going to do to Misha?
Well, we've done one thing to him already. A willing volunteer. He's my son. He had no option.
So you say willing.
You're willing. A very willing subject. So we've put some drops in one of his eye because we need to open up the window a bit. Okay? Mm-hmm. And we need to get a lot of light into the back of the eye to show the region which allows you to see us and this lecture this evening, which is known as the macula.
The macula?
Yeah.
Okay.
So he's going to view through Misha's clear window. It gives us a complete view.
And we can see his pupils dilated there, can't we?
Yeah. It's not that he just is enjoying himself. But, so if you want to go through that window, Nick, and take us to the back of the eye. Yes, here we go. So this is the optic nerve head. That white disc is where all the information that's processing the light and vision leaves the eye and goes to the brain. Mm-hmm. And if we go slightly to the side of that, Nick, this area, this little dotted area here, is that macular region. And that contains all the information and all the cells to allow us to see the world.
So what goes wrong with the macula sometimes?
So that macular region contains these cells. This was taken from a brand new piece of equipment. Again, Misha kindly volunteered yesterday
So this is Misha's eye again?
This is Misha's eye again.
But we're honing down even more and we're looking at the actual photoreceptors themselves.
These are no bigger than a pinhead, and these are all the colour receptors, the cones that actually give us that high, what we call high visual acuity. Cheers, Mish. And in fact, Mish was the first person under 21 in the UK to get this image of his eye.
Wow.
And this technology comes from the Hubble telescope. So you need this to have healthy vision. This, sadly, is what happens to people over the age of 65, and it's called age-related macular degeneration.
Does anyone in the audience know somebody that's got age-related macular degeneration? People in the gallery have got their hands up, yeah. It's a generation thing, isn't it?
It's often called Alzheimer's of the eye because the numbers are huge. People over the age of 65, nearly a quarter of them have some form of this disease.
And what goes wrong in this disease?
The one beauty of the eye, if we actually put blue light into Misha's eye, this isn't Misha's eye, then those support cells which are behind the seeing part autofluoresce, they glow. And as you see, we have this massive hole
in it. Yeah.
And those cells have died. These are the support cells. These are what keeps your seeing eye healthy. So anyone under the age of 65, hopefully everything glows. But people over the age of 65, a number of them start to see those cells die. And this is the macula, right over the macula. So this person would not be able to see.
So what does that actually feel like? And we can actually see what that might feel like, what sight might be like with age-related macular degeneration with a volunteer. Can I have a volunteer, please? We'll have you there in the pink. Thank you. If you'd like to come out. What's your name? Isabel. If you put these glasses on, Isabel, that will mimic what it would be like with age-related macular degeneration. Is that weird?
Yeah.
Can you read the top line?
No.
If you can't read the top line, you would actually be determined as being blind. Okay? Can you just move your head to the side and try and use the side of your eye?
Does that work a bit
Keep
better?
going. Yeah. Can you start
Yeah.
reading down?
C,
See, yeah, good. Next line. Can you see any letters?
S,
Good. Next line.
V,
Oh, you're doing really well.
K.
Okay.
But you can't see now if you look centrally, if you look full on. So it's the middle of your visual field that's very difficult to see. I think we better relieve you of those glasses now, haven't we? We can all have a look at what this looks like. If I bring this slightly odd-looking plate to the camera lens here. Yeah, can we see that? So that's what happens when you have age-related macular degeneration. The central part of your field of view is the thing that goes very blurry. So this is a pretty easy condition to diagnose, I guess. So the big question is, what's the plan?
So those cells, those middle layer cells that are called retinal pigment epithelium, you're born with and you die with. They don't, as you were saying earlier, some cells don't regenerate through your life.
So these are the retinal pigment epithelial cells.
They are indeed.
They're the cells that nourish that whole region of the macula.
They do.
And they wear out and they can't be replaced. And that's what causes age-related macular degeneration.
It is. So the only way of actually helping people who suffer from this disease is if in some way we can put new RPE, retinal pigment epithelial, back in that area where they've died. And that's exactly what we've managed to do. By taking an early stem cell, which we're going to show you now, we can actually see whether we can turn a stem cell into an eye cell. So Bias is just going to focus in on the cells. There's two characteristics of the cells. The first characteristic is they are black. And that's because they have pigment granules. And in fact, that's why you've got the black component in your eye. And the second one is this crazy pavementing. And we actually call that cobblestone morphology. And this is a really characteristic of these cells. It's very unique of these cells. So we now have the opportunity to replace the area of the macula in which those cells have died.
So you've grown up lots of these cells now in the lab, and I understand that these cells are going to be used in a very early clinical trial to try to help patients with age-related macular degeneration.
They are indeed. They now have given us approval to start a clinical trial next year.
And this is the first time that this number of RPE cells have been grown up in sufficient quantities to actually do this kind of a trial. It's the first time it's happened in Europe. So we're really, really lucky to be able to see these cells this evening in the lecture theatre. Thank you so much, Pete.
Pleasure.
And Bianca. So these are new treatments in progress right now and from which you'll benefit from in the future. Some of these new therapies might seem like science fiction, but it's happening now, and it'll be part of the world that you grow up into. So what about this living forever business? How will all this knowledge help us live healthier lives for longer? So we've heard a lot about cells and how cell therapies might help to cure age-related diseases and other diseases, of course, as well. The more we understand about our own genetic makeup, the more we can personalise our medicine. And to help us understand the medicine of the future a bit more, ladies and gentlemen, please welcome Robert Winston. Good to see you. Thank you for coming.
Well, Alison, got some bad news, really, I'm afraid. At Imperial College, where my lab is, we spent £10,000 and we've sequenced your entire DNA,
my DNA?
all 3 billion letters of your DNA alphabet.
And what did you find?
And here it is. And you see that there are some dodgy bits, actually, there.
Dear.
And I think you're now 72? Oh.
Do I look that old?
Well, let's say you're. We calculate from this sequence that you're going to die in 2053 in July.
Oh my goodness. Is that what it's going to be like, going to the doctor when they've got your DNA sequence? Would I want to know when I'm going to die? Would you want to know? Yes! Who said yes? Who said yes? Well, why would you want to know?
I would want to know when I was going to die because then I could kind of plan ahead.
Plan ahead. Well, you might think that when you're your age, but when you're my age, and when you're Robert's age, would you want to know?
Well, actually, every day I live, I have another 6 hours added to my life. Did you know that?
So you're on a roll.
And that's the same for all of us, actually. But the interesting thing, of course, is that this is fortunately very imperfect. Even in my university, like Oxford, we're not absolutely perfect. So,
no, we're not.
So
could this be useful, this sort of information?
To tell you the truth, Alison, I'm pretty dubious. I think that there's a lot of talking about the genome, DNA, to really detect and predict our diseases. And I think, I'm sure you probably agree with me, apart from the genetic disorders, the specific single gene defects that some of us have, it's going to be very difficult to deal with heart disease, diabetes, the really big diseases, I think, are going to be a very big problem.
Because they're so complex.
And because they have so many genes involved,
Yes.
or partly involved, and other things, environmental effects on genes too, that we're beginning to understand
Absolutely.
about.
But so do you think we could ever live forever?
No.
Would
you want to?
Would anyone want to? Would anyone here want to live forever? Yes, what about you up there in the red?
Make fun of people who don't live forever.
You might be a bit lonely in the end, mightn't you? But you certainly wouldn't want to live forever.
Well, the predictions are, aren't they, that we probably will have. I mean, most of the people in this audience will live to at least 90. Probably, maybe as many as half might live to 100. Mm-hmm. But we're still going to have a finite lifespan, don't you think? I mean, like your CL.
Oh, I think it's I think living forever is
not a good idea.
Is not on the near
also,
horizon.
of course, we can't avoid the mutations which build up with every cell division.
Absolutely.
So we're going to be more and more prone to all sorts of rather unpleasant disorders.
Absolutely. Robert, thank you so much
Pleasure.
for coming and talking to us this evening. Thank you so much. So if we go forward with cell and gene-based therapies then, are there unforeseen consequences? Could there be a problem? Once you've edited a genome, for example, what are the consequences of doing that? We could be permanently changing our genetic destiny. We could be at the dawn of a new era in medicine, ladies and gentlemen. We have remarkable opportunities to alleviate human suffering at the hands of disease on a grand scale, as we've seen. We mustn't misuse this knowledge. The decisions of the kind we have considered will need to be made in the future by you and your children and your children's children. We're all owners of this knowledge together. But we will not be able to make good decisions if we don't all understand the science behind these issues. The next chapter of Life Fantastic is for you to write, and it's the most important chapter of all. Thank you and good night.